The central dogma of molecular biology, which was first proposed about 70 years ago by Francis Crick, holds that DNA, which contains all the information needed to build a cell, is transcribed into RNA, as you see single-stranded, which is then translated into the structures of all the proteins that make up the cell. DNA is also replicated so that this information can be carried to the next generation. The DNA is a double-stranded polymer, very long, that's composed of nucleotides or bases. We abbreviate them by the initials of their names, A, T, C, and G. You see here a strand, one of the two strands, with the sequence of the nucleotides shown. The basic rule of molecular biology is that those four nucleotides pair with each other in very specific ways. The A always and only pairs with a T, or in the case of RNA, a U, and the C always and only pairs with a G. This rule is critical for understanding all parts of the central dogma of molecular biology. What the rule means is that if you know the sequence of a particular strand of DNA or RNA, in this case we're showing RNA, you automatically know the sequence of the other strand, which is called the complementary strand. So for every A, we have opposite a U. For every G, there's a C. For every U, or in the case of DNA, a T, there's an A. And for every C, there is a G. Getting back to DNA, we find that it's more convenient to write these sequences in groups of three. Every group of three nucleotides is called a codon, and the codons are the words of the language of DNA that give the information to the RNA and then to the protein synthesis system. Each codon consists of three nucleotides, or three letters. Here we see the genetic code, which shows what each possible combination of the three nucleotides codes for. If you look, for example, at the circle, you'll see that there are four different combinations of nucleotides that code for the amino acid threonine. They all start with AC. Every possible combination codes for an amino acid, except for three of them which code for stop. So here we see that, for example, the codon ACT, which is written in words in the bottom, means put a threonine next in the protein you're building. On the top you see the chemical version of ACT, adenine followed by cytosine followed by thymine, and that means the amino acid threonine, which is shown in its chemical structure. What's important to understand is there's no chemical basis for the coding system. In other words, the codon does not interact in any way with its amino acid. So the assignment of each codon to its amino acid is arbitrary. If we try to find some way that the chemical threonine would react with the three bases that form the codon that codes for it, we find that there is in fact no such interaction possible. The first part of the process to make proteins is to transcribe the information from the DNA to the RNA. That's done by an enzyme called RNA polymerase. And what happens is one of the two strands of DNA is copied into the RNA. But what you'll see here is that for every A, we get a U, and for every C, we get a G, and so forth. In other words, the RNA contains a complement of one strand of the DNA, which means it's identical to the other strand of the DNA. That RNA that's produced is a single strand. Once it's made, it leaves the DNA, it leaves the nucleus, and goes into the cytoplasm. This is a slide showing an oversimplification of the entire process of protein synthesis. You see, again, the messenger RNA, which is the long red stripe, that goes from the DNA into the cytoplasm where it finds a structure called a ribosome. And there, with the ribosome and with another kind of RNA called transfer RNA, the protein is made. We're going to go into some detail as to how that happens. One of the key enzymes in this process is called amino acyl tRNA synthetase. There are 20 of those, one for each of the 20 amino acids, and they are very complex enzymes with a whole range of functions. They have to bind a particular amino acid and activate it with ATP. They also have to bind several cognate tRNAs, the ones that fit the correct amino acid, and that's done based on the shape and the anticodons on the tRNAs. They have to edit the amino acids to make sure they're specific. They attach the amino acid to its correct tRNA, and each one uses a different strategy to accomplish all this. I'm now going to show how the AA tRNA synthetase works. Here we see in blue the enzyme. Of course, it's a schematic of the enzyme alanine tRNA synthetase. Its job is to attach an amino acid, alanine, to the correct tRNA for alanine. The first step is to bind the amino acid to a specific binding site, which is made to precisely fit the amino acid alanine. The enzyme also contains binding sites for the tRNA, in this case the alanine tRNA. It binds both according to the shape, and you see that with the arrow-shaped part of the tRNA, and with its anticodon. The base of each tRNA is an anticodon that fits the codon for that amino acid. The next step is the enzyme produces a bond between alanine and its tRNA. That is a covalent bond, so it's very strong. Now we have the correct amino acid, alanine, covalently bound to its transfer RNA, which contains the anticodon, which will bind to the codon for alanine. But let's go back a little bit. Before that happens, the enzyme does something else. It takes the alanine, it takes the amino acid bound to its binding site, and moves it to another site, which is that open round site right next to it. And what happens is here the alanine does not fit because that other site, it's called the editing site, is too small for the alanine to get into it. And in that case, what happens is the alanine then goes back to its bound site and waits for the tRNA to come in and then gets bound to its tRNA. So the editing site has no effect on the alanine. On the other hand, let's say a different amino acid, a smaller one, in this case glycine, mistakenly binds to the site on the alanine tRNA. In other words, it's the wrong amino acid, but it does fit into the binding site, not perfectly, but just enough to bind. Just as before, the enzyme moves the amino acid that is bound to the amino acid binding site into the editing site. But this time, the amino acid glycine, which is smaller, fits into the editing site. And once it fits into the editing site, it is destroyed. This is one way that this enzyme, the amino acyl tRNA synthetase, is able to ensure binding of the correct amino acid to the correct tRNA. So now we have alanine attached to the alanine tRNA with its anticodon GCA. Now the codon for alanine is CGT. In the messenger RNA, it will be represented as CGU. And so we now have a binding between the codon and the anticodon. So the original codon in the DNA was GCA. It was copied into the mRNA as CGU, and we now have binding between that codon and the anticodon. So we now have alanine in the correct position. Let's take a look at more detail in how this works in actually building proteins. The oval circle represents the ribosome. The black bar is the messenger RNA, and we see there are 5 codons represented here. The first step is that the first amino acid in the protein chain, in this case methionine, bound to its tRNA with its anticodon, will go into the ribosome, and as you see, it has a match between its anticodon UAC and its codon AUG. The second amino acid, glycine, attached to its tRNA goes into the second site, and again we have the GGG codon and the CCC anticodon that match. And base pairing occurs between the codon and the anticodon, so this is now stable in the ribosome. The ribosome then makes a covalent bond between the methionine and the glycine, the first 2 amino acids. This is called a peptide bond, and it binds these 2 amino acids together. This is the beginning of the new protein. What happens next is that the ribosome moves 1 codon along the messenger RNA chain so that the methionine tRNA is now outside of the ribosome, and there is an empty spot for the 3rd amino acid to come in. Once outside the ribosome, the base pairing between the codon and anticodon for methionine, the first amino acid, is broken, and the tRNA for methionine leaves the system. Now the 3rd amino acid in the protein chain, valine, attached to its tRNA with its anticodon, CAG, which matches the codon GUC, comes into the empty site. Again, base pairing between codon and anticodon occurs, and again, a peptide covalent bond is produced between the 2 amino acids in the ribosome. In this case, it's now the 2nd glycine and the 3rd valine. And again, the ribosome moves 1 codon away, leaving an empty space for the 4th amino acid to arrive and be bound to the valine. And this continues throughout the process so that every codon on the messenger RNA is eventually matched with the anticodon of the tRNA containing the proper amino acid that belongs in the next position on the protein chain. And eventually we have a long chain of amino acids, all connected with each other in a straight line. And here we see 8 amino acids, but in reality, proteins usually have anywhere between 100 and 300 amino acids. Some are even larger. Once a stop codon enters that ribosome, that ends the synthesis of the protein. The protein is then released, the messenger RNA disappears, is degraded, and we're left with a protein chain. But most proteins are not long, straight chains like this. They tend to be folded in all sorts of shapes because most of them are enzymes, and their chemical activity, which is so important for the function of the cell, depends on its being in the right shape. So proteins are folded into a very specific shape once they leave the ribosome, and at that point they are able to perform their functions in catalyzing chemical reactions or whatever they're required to do. Here we see 3 images of proteins showing different ways that they're depicted, but they all show that these have shapes that are not long and linear, but various kinds of shapes. Everything you've just seen is part of how it is that we are able to have such high accuracy self-replication of cells. The final accuracy is 99.9999%. Now this leads to an enigma, a biological enigma. The genetic code, the ribosomal translation system, which you've just seen, the aminoacyl tRNA synthetases enzyme, are all necessary for accurate self-replication, and that is necessary for evolution. So how could all of those systems evolve since they are all needed for evolution? That's a question that is not resolved, and may be very difficult to resolve. Thank you for watching this video, and stay tuned for more videos in this series. Also, please hit the subscribe and like buttons, and as always, questions and comments are welcome. God bless you all.